Hello and welcome to another episode of Pesticide History. In this episode we'll cover the history of herbicides. We'll start with the history of non-selective herbicides, which are herbicides that kill pretty much everything, followed by selective herbicides, which only have effects on one broad group of plants, such as monocot versus dicot, or perennial versus annual. Since this is an agricultural channel, mostly anyways, it will only cover herbicides used in agriculture or plant production. We'll start with Paraquat and Diquat. These are both bipyrimidium-based herbicides. Diquat was first created in the late 1950s, and the former was created in 1962. Paraquat persists a lot longer in the soil than Diquat, and both are very toxic to humans and really all mammals. The mode of action of Paraquat is basically chemical lands on the leaf and reacts with sunlight, creating free radicals. The free radicals burn through the cells and eventually kill the plant. As of right now, Paraquat is not used very much in the Western world, but it's still used in developing countries. Now on to Glyphosate. It was originally marketed as a cleaning agent, but it was soon found that it was really useful for killing weeds. Its mode of action is that it inhibits the ESPS enzyme within plants, both preventing the creation of different amino acids that the plant needs to survive, and creating free radicals that damage a plant even further. These two processes eventually kill the plant. Now on to glufosinate. Glufosinate was created in the 1970s by combining left-handed and right-handed versions of phosphine oricin. This chemical prevents the synthesis of an important plant chemical called glutamine, which kills the plant. Like glyphosate, it is non-selective and kills all plants equally, so genetic engineering was used to make crops immune to said pesticide in order to make it suitable for agriculture. In the case of glyphosate, there's many different ESPS enzymes, and such enzymes are found in fungi, bacteria, and plants, but not in animals. Hence why we are not affected by glyphosate in the same way plants are. One bacterial species, however, had an ESPS enzyme that was not affected by glyphosate. The gene that codes for this version of the ESPS enzyme was then used to replace the genes within corn and soybean genes for ESPS, thus granting immunity to this herbicide. In the case of glufosinate, a species called Streptomyces had developed interesting genes called BAR or PAT genes that can detoxify phosphophenorycine. Those genes were encoded into the plant through genetic engineering, granting immunity to glufosinate. Now on to selective herbicides. In the 1940s, researchers discovered the plant hormone IAA, which controls plant growth. It was also discovered that high concentrations can stop plant growth or create irregular plant growth that can kill the plant. However, it only works on broadleaf plants, making it suitable for use in fields that grow grass-based crops, such as corn or wheat. So researchers began looking for more powerful analogs of IAA. The first discovered one was MCPA, which wipes out all broadleaf plants within a field, leaving only grasses such as barley and oats, the actual crops. A derivative called MCPB was safer in that it did not work on legume plants. A later version called 2,4-D, made in 1945, was also created. It was called 2,4-D, and it works in much the same way, but at a more potent level. Due to a volatility issue, a new version called 2,4-D-C was made to be less volatile. This, in combination with semi-dwarf wheat varieties and fungicides, increased wheat yields by a considerable margin. A later derivative of 2,4-D called 2,4,5-T was developed to deal with woody weeds, and was first used in Agent Orange in combination with 2,4-D to destroy forest cover during the Vietnam War. However, it was found that there was a chemical contaminant made as a result of the manufacturing process for 2,4,5-T, and this contaminant caused cancer. This chemical is called TCDD. Now on to Atrazine. Atrazine inhibits photosynthesis at photosystem 2, one of the two systems used to produce photosynthesis. Corn, however, can detoxify this herbicide, making it useful for controlling weeds within a cornfield. Next is Dicamba. Dicamba was first found during the development of 2,4-D as an offshoot of the research. It was, however, far too volatile to be used. With the advent of brand new herbicide-resistant weeds, such as Canada fleabane, new herbicides to kill these plants were needed, resulting in Dicamba being released. Due to the volatility, it needs to be used a lot more carefully since it can damage non-resistant crops downwind from the spray very easily. And various technologies to combine them in different formulations, such as the vapor grip technology, were made specifically to make a less volatile version of Dicamba. Now on to our final herbicide, the sulfonylureas. This group of herbicides are acetolactase or ALS inhibitors. By inhibiting the ALS chemical, the plant is unable to make the amino acid valine, isoleucine, and leucine. These three are necessary amino acids to make DNA. Without these amino acids, no more DNA can be made and the plant dies. This specific chemical that works on ALS only kills broadleaf weeds that are annual in nature, although there's other ALS inhibitors that work on grass weeds as well. Well, that about covers everything. Thank you for watching. Thank you for watching this video. If you're watching on YouTube, please subscribe to me on BitChute for a greater variety of content, 4 videos a week. And thank you to all my subscribers on both platforms. I appreciate it.